Hello friends, Ace Botane next episode. In this episode, we'll be discussing NCERT Cook reference. Under that series, Respiration Plan's 9th episode. I remember daily I'm putting 2 questions in Ace Botane for NEET exam around 12:45. So subscribe Ace Botane, so you'll be getting all this detailed note already I published in YouTube under Ace Botane. This is for quick reference, theory and NEET. So for convenience, I will share the screen because time is important for us. So I will share it. I think a screen came, so we can proceed. All references based on NCERT March 2024 repeated edition. See, assumptions that is starting with it is possible to make calculations of the net ATP for every glucose molecule. So possible. That is what NCERT says. Possible. Okay, possible. Net of the net ATP for every glucose molecule release. But in reality, this can remain only a theoretical exercise. These lines can be made into a NEET type question and theory question like fill in the blank. These calculations can be made only on certain assumptions. NCERT page number 161 is given. And assumptions, 4 items they're given. What they're telling is there is a sequential orderly pathway functioning, like from one substrate to next, with the pathways like glycolysis, TCA cycle, ETS. NADH is synthesized in glycolysis. That NADH enters into mitochondria and enters into oxidative phosphorylation. None of the intermediates in the pathway are utilized to synthesize any other compound. None of the intermediates. That's what NCERT says. Only glucose is being respired. Another assumption. No other alternative substrates are entering into the pathway at any of the intermediate stage. So with these 4 assumptions, we are proceeding. NCERT only, we can continue. I think first page is clear to all of you. Now, second page when we enter. But this kind of assumptions are not really valid in living systems, because this type of reactions are taking place even simultaneously, just like when we study mitosis and meiosis. We are sequentially studying prophase, anaphase, metaphase, telophase likewise, or prophase, metaphase, anaphase, telophase. This way proceeding. That is sequential manner. But different cells, different stages we can observe, same manner. ATP is utilized when needed. Whenever there is a need of ATP, ATP will be utilized. Enzymatic rates controlled by different manner. But these all are beautifully and efficiently managed. So there is a system to manage all this, suitable to living system. That's 38 ATP molecule in aerobic respiration per 1 molecule of glucose. This is what NCERT says. But when we compare with fermentation, in fermentation only partial oxidation of glucose. So that's less ATP only, that is 2. 2 only finally produced in fermentation, anaerobic respiration, net is 2. NADH is oxidized to NAD+, rather slow in fermentation. Slow in fermentation, but in aerobic condition it is very vigorous. NCERT page number 161, 62. Sorry. I think this page is clear, so we can proceed to calculation part. This is a basic calculation based on the data. Oxidation of 1 molecule of NADH can provide 3 molecules of ATP. 1 molecule of FADH provide 2 molecules of ATP. That is NCERT repeated edition March 2024, page number 160, it is given. Now with this we are starting respiratory stages, source, and number of ATP. Glycolysis in cytoplasm direct way, it is 2 ATP. Glycolysis in cytoplasm with 2 molecules of NADH, that is each NADH2 can normally provide 3 ATP. In that way, it is 6. So total 2 plus 6, 8. Now link reaction, that is pyruvic acid to acetyl coenzyme A, 2 molecules of NADH2 will be produced, because 1 pyruvic acid, 1 link reaction, second pyruvic acid, second link reaction starting from 1 molecule of glucose. So 2 more NADH2, that means each NADH2 3 ATP, so 2 into 3, 6. So it will be 6. I think after that is clear. Now Krebs cycle or TCA cycle or citric acid cycle, 6 NADH2, 2 tenths of Krebs cycle, because 2 link reaction, 2 pyruvic acid, 2 Krebs cycle, 6 into 3, 18. And there we can also find 1 FADH2 in 1 Krebs cycle. So 2 Krebs cycle, 2 FADH2, 2 into 2, 4 ATP. Then next is substratal phosphorylation, that is direct 2 GTP. 1 in 1 Krebs cycle, other in another Krebs cycle. So 2 Krebs cycle, 2 GTP. It will be 2. So total will be 14. 14 plus 18, 32, 36, 38. I think idea clear. Can be 36 based on glycerol phosphate shuttle, or with the 2.5 and 1.5 ATP respectively. If we calculate, then value will be different. But this is a common energy calculation. I think idea clear. If you like this episode, subscribe Ace Botane, like it, share it, comment on it. We can continue the series. We'll be meeting later. So take care. Bye. Thank you.